All right. So let's talk about mRNA structure, translation, and stability in this lecture. So in this lecture, we will cover the basics of mRNA with a focus on what actually matters for modeling. Of course, we will start with the physical structure of a typical cellular mRNA, what it is made of and how it is organized. Then we will look at the natural ranges of key properties such as length, folding energy, and half life. We will see how translation and decay work, and we will talk about untranslated regions and their importance. We will also introduce circular RNAs since they behave differently from linear mRNA. You definitely need to know this. The goal here is to give you a baseline. Let's start with the big picture. A DNA holds the genetic code, but DNA stays in the nucleus and it doesn't come out. So, how do we make proteins? We need a messenger molecule. That's what we are going to talk about today. RNA polymerase reads a section of DNA and builds a messenger RNA transcript. So this is actually a pre-mRNA. That transcript gets processed. Introns are spliced out. A cap is added, and then a poly A tail is added to 3' end. Now you have a mature mRNA. And then this mRNA exits from the nucleus to cytoplasm through a nuclear pore. In the cytoplasm, ribosome translates it into protein. So the key point for us is therapeutic mRNA skips all of this because we deliver finished mRNA. So that's the focus for us in how to model that molecule. Um, the thing is, mRNA is a polymer. That just means it is a long chain built from repeating units. Those units are called nucleotides. And for RNA, there are only four nucleotides, right? A, U, G, C. Inside the cell, those come from ATP, UTP, GTP, and then CTP. I mean, that's not super important right now. But in any case, each nucleotide has three parts: a base, a sugar molecule, that is ribose, and phosphate. So the sugar and phosphate form backbone. And now one thing matters for everything downstream, that is direction. So RNA runs from 5' to 3'. Translation, decay, folding, all of it depends on that directionality. I mean, if you forgot this 5 prime, 3 prime, that's basically, this is the ribose molecule. And then this is carbon number 1, 2, 3, 4, 5 prime. Just to differentiate other carbons from the base, you have prime. So this is 5 prime. And then this is 3 prime. We need to understand few things about RNA molecule folding. We already know, G pairs with C and A pairs with the U. Here you can see that. But one thing is a G to C is 3 hydrogen bonds, whereas A to U is 2 hydrogen bonds. So GC rich regions hold together tightly, whereas AU rich regions are a bit more flexible, a bit more accessible, and there is one more layer you need to know. That is, these bases not only just pair because of this secondary structure, they also stack along the backbone. Those stacking interactions depend on local sequence context. Some combinations pack a bit tightly, others maybe not. So what we need to know here is that yeah, we know that sequence encodes whatever is going to be formed in some protein, but not only that, the sequence encodes the structure of the molecule itself. So the GC content and then the local context, they drive folding and then the stability and how accessible the RNA is to ribosomes and enzymes. So RNA folds into stems, loops, and hairpins through base pairing. To describe how stable those folds are, we use minimum free energy, or simply MFE. So this MFE comes from folding algorithms. It estimates the energy of the most stable predicted structure. More negative values mean tighter, more stable folding. So values closer to zero mean the RNA is a bit loose and a bit more accessible. In natural mRNAs, small stem loops are often in the range of about -5 to -15 kilocalories per mole. Larger structures can reach -15 to -30. Um, Near the 5 prime cap, structures are typically much less stable. So, I mean, that means the region is kept relatively open to allow ribosomes to bind and start translation. For modeling, the key idea is not to optimize MFE globally, but to understand how local folding affects accessibility and function. Now, let's talk about codons. mRNA is not uh, read letter by letter. It's read in blocks of 3 nucleotides. And this is what a codon is. Um, so for each codon, a matching tRNA molecule arrives and delivers a specific amino acid. That's how a protein chain gets built, see? how the amino acids are being added. So what we need to understand is that um, the reading frame matters. If you shift by even one nucleotide, everything in the downstream is just wrong. Another important thing is codons affect translation speed, accuracy, and how much protein you actually get. So there are 64 different codons, 61 specify amino acids, and 3 are used as stop signals. But remember, we have only 20 amino acids, so there is obviously redundancy. AUG, that is a start codon, and brings methionine molecule. That's the beginning of a protein. Whereas UAA, UAG, UGA, these are stop codons, and that marks end of protein. Just one thing you need to remember, just as an example, glycine is encoded by GGU, GGC, GGA, GGG. So these are called synonymous codons. So these synonymous codons are not functionally equivalent. They differ in translation speed. We can change codons without changing the protein, but we do change translational kinetics. And now let's zoom in on translation itself. So the ribosome is essentially a molecular machine that moves along the mRNA molecule from the 5' end to 3' end. So the ribosome links those amino acids together, because tRNAs bring amino acids based on the codons, right? We just saw that in the previous slide. So from a modeling perspective, the important point is this. Translation is sequential and local. What happens at one position depends on the nearby sequence, structure, and timing, not just the final protein sequence. So that's why features like codon choice, secondary structure, and ribosome spacing matter, even though the genetic code itself is fixed. Before we continue, let's summarize what we have covered. In the nucleus, DNA is transcribed into pre-mRNA. The transcript gets processed, introns are removed, and the 5' cap and poly A tail are added. Then the mature mRNA is exported. Once it reaches the cytoplasm, ribosome binds to it. So that's a 5' cap, right? It reads the sequence from 5' to 3', and one codon at a time, building a protein by adding one amino acid per codon. The result is a protein chain. So typical human coding sequence is, something like 1200, 1300 to 1500 bases. That gives you a protein of roughly 400 to 500 amino acids. I mean, that's just common. Um, so what we are learning here is that we are going to design this mRNA molecule. Now let's look at the full architecture of mature mRNA molecule. I think it is helpful if we think mRNA as a modular system. There are five functional regions arranged from the 5' end to the 3' end. Um, at the very front left, um, you know what I mean, is the 5' cap. It is a modified guanosine that protects the RNA and recruits the translation machinery. Proteins like IF4, so these molecules bind and ribosomes load onto them. Next is the 5' UTR, that is untranslated region. This region is not translated like the name tells you, um, but it strongly influences how easily ribosomes can initiate. Its sequence and structure matters a lot. Then comes the coding sequence. So this is the part that actually encodes the protein, and then the codon by codon. After the stop codon, we have 3' UTR. Again, it's untranslated region, so doesn't code for anything, but it is a major regulatory hub. It binds factors that control stability, localization, and translation efficiency. Finally, we have poly A tail, a long stretch of adenosine molecules. So one thing here is it is added enzymatically, not encoded in the genome. Proteins like PABP bind here and help stabilize the mRNA and boost translation. So the key point is that these regions don't act alone. CAP, UTRs, poly A tail, they all work together as a coupled system. That's what determines how much protein you actually get. Let's talk about the 5' cap a little bit more. Structurally, the cap is a 7-methyl guanosine linked through a 5' to 5' triphosphate bridge. A natural mRNAs differ mainly by a methylation state. If there is hydrogen, that is cap 0. If it is a methyl group, that's cap 1. If you have methyl group here also, that is cap 2. So this is nucleotide number 1 and number 2, and then it goes on, and then you have 5' bridge to 7-methyl guanosine. Um, Functionally, so just one point is cap1 is dominant in humans. Functionally, cap does three things that we care about as engineers. First, it recruits the translation machinery. Initiation factors binds the cap and provides entry point for ribosomes. Second, it protects the RNA from degradation by blocking 5' to 3' exonucleases. Third, it signals immune self-identity. And now let's talk a little bit about 5' UTR. So we can think we can think of this as a control region for translation initiation. It strongly determines whether ribosomes can even get started. In human mRNAs, the 5' UTR is typically tends to a couple hundred nucleotides long. So that is between cap and AUG start codon. One thing here is, GC content varies, and that variation obviously matters. Higher GC promotes secondary structure, which can slow ribosome scanning, whereas lower structure generally makes initiation a bit easier. So, one thing here is that the ribosome does not start at just any AUG. It typically looks for an AUG in the right sequence context, that is the Kozak consensus sequence. Strong context means efficient initiation. Another important feature is upstream open reading frames, UORFs. If the 5' UTR contains extra AUGs before the main start codon, ribosomes may initiate early and never reach the intended coding sequence. So what we need to know from this slide is 5' UTR controls accessibility, scanning, and start codon recognition. Now, let's talk about the coding sequence itself. So this is the part of the mRNA that actually makes protein. It starts with AUG, start codon, and ends at a stop codon, that is UAA, UAG, or UGA. Uh, Two things matter here. First, what protein is made. So that is obviously defined by the amino acid sequence that was encoded by the codons. Second, how fast the protein is made. That is controlled by translational kinetics. Common codons match abundant tRNA, so the ribosome moves quickly. Rare codons rely on scarce tRNAs, so the ribosome slows down or pauses. GC content also matters. High GC tends to form stronger secondary structures, as we discussed in previous slides, which can physically slow ribosome movement. Lower GC generally means weaker structure, so faster translation. Okay, now let's talk about 3' UTR. So this region sits between the stop codon and the poly A tail, and typically it is much longer than 5' UTR, hundreds to thousands of nucleotides. So functionally, 3' UTR controls three major things. First, stability, how fast the mRNA decays. Second, translation efficiency, how much protein gets made from a given transcript. And third, subcellular localization, where in the cell that mRNA ends up. So much of this control comes from specific sequence motifs. AU-rich elements tend to recruit de-adenylation machinery and drive rapid decay. MicroRNA binding sites can repress translation or actively trigger degradation. So, I mean, the key point here is that 3' UTR is extremely diverse across transcripts. So this is where most of the regulatory information lives. Oof, all right. I think finally we reached to poly A tail. Um, Structurally, newly synthesized mRNAs leave the nucleus with a poly A tail of about 200 to 250 nucleotides. So the tail is coated by poly A binding proteins. The poly A tail has two primary functions. First, it enhances translation by interacting with the EIF4G, effectively linking the 3' end back to the cap. Second, it protects the transcript from exonucleases. Over time, deadenylases gradually shorten the tail. As the tail gets shorter, PABP molecules fall off. Once that protection is gone, exonucleases can access the mRNA and degradation proceeds. So the key idea is, we can think that the poly A tail acts like a timer. It shortens steadily over the lifetime of the mRNA, setting how long that message remains protective. Let's summarize translation. So translation has three phases. The first one is initiation. Here, the small ribosomal subunit binds the 5' cap and scans along the mRNA until it finds an AUG start codon. Once that start site is recognized, the large subunit joins and the ribosome is fully assembled. And next is elongation. The ribosome moves along mRNA codon by codon in the 5' to 3' direction. So each codon is decoded by matching tRNA and an amino acid is added and see the protein chain grows and then finally termination. When the ribosome encounters stop codon, release factors bind and then the protein is released. From a rate perspective, two steps usually dominate. Initiation is limited by how easily the ribosome can be recruited and scan the 5' UTR. Elongation is limited by tRNA availability and codon decoding speed. So overall, translation speed depends on how efficiently the ribosome starts and how smoothly it moves. Let's see natural mRNA stability. In cells, mRNAs do not all last the same amount of time. Their half-lives span more than an order of magnitude, and that range is functionally important. On the short end, some mRNAs last only about 30 minutes to 2 hours. These typically encode proteins like cytokines, transcription factors, or immediate early response genes. They usually contain AU-rich elements in 3' UTR, which actively recruit deadenylation and decay machinery. In the middle range, change roughly 2 to 8 hours are many metabolic enzymes and housekeeping genes. So these transcripts strike a balance, right? So they are stable enough to support ongoing function but still responsive to regulation. On the long end, some mRNAs persist for 8 hours, a full day, or even longer, even months. So these often encode structural proteins or very abundant housekeeping functions. So what distinguishes these long-lived transcripts is its control of deadenylation. The rate at which the poly tail is shortened varies by roughly tenfold across transcripts, and that rate largely determines mRNA lifetime. So far, we discussed about linear mRNA, which is built to be translated efficiently but decays on a predictable timescale. So circular RNA is very different design. In circular RNAs, 3' end is covalently joined to the 5' end. There are no free ends. So that single structural change makes them highly resistant to exonucleases. As a result, circular RNAs often persist for hours to days, much longer than typical linear mRNAs. But that stability comes with a trade-off. Because circular RNAs lack a 5' cap and the poly A tail, so they don't support normal cap-dependent initiation. So most endogenous circular RNAs are translated very poorly, if at all. So functionally, cells mostly use circular RNAs for regulation rather than for protein production. Things like microRNA sponging, scaffolding protein complexes, or modulating translation of other RNAs. You will also notice they are especially abundant in brain and immune tissues where long-lived regulatory signals matter. Um, Important thing is when circular RNAs are translated, it is usually through non-standard mechanisms, that is internal ribosome entry sites, IRES, or M6A mediated cap independent initiation. So the key points from this slide is circular RNA trades translation efficiency for extreme stability. It lives in a very different part of the design space than conventional mRNA. Let's summarize what we learned in this lecture. We discussed chemical foundations about nucleotides, base pairing, secondary structure. We discussed about the 5 regions of mRNA, cap, UTRs, coding sequence, and then poly A tail. And then we learned that each contributes to translation efficiency or stability. And then we discussed a little bit more about translation, and then we discussed about initiation, elongation, termination, and then we discussed about 3' UTR driven regulation, and then we discussed about circular RNA, that is because there are no free ends. It is exonuclease resistant, but it requires cap independent initiation. So overall, what we need to understand is the mRNA molecule is a modular system one can design by modifying nucleotides systematically for therapeutic purposes, I mean.